So today I am talking about high throughput sequencing and just kind of like the how it works. I'm going to be focusing on. See, I was okay until I drank the tea. Okay, how it works. I'm going to be focusing on Illumina for a lot of it, but I'm going to touch on some other technologies towards the end, and actually we'll start off talking about Sanger sequencing. And this is just going to be me, unfortunately for me today, this is just going to be me talking for the next like 3 hours. I'm planning on taking a break like every hour or so, just so I have a break and you all have a break. Feel free to ask questions, interrupt me, whatever. I think I can hear people on Zoom, and I can see the chat, but there's like, if you were here, you'd see there are like 20 million screens. So if you put something in the chat and I don't see it, you know, you can put it in the chat again, or you can just unmute and yell. Okay, any questions before I get going?
I have one.
Yep.
Yes, so the on DCC on demand, are there new materials?
So there's not going to be anything on, we're not going to be using DCC on demand at all today.
Oh, okay.
So for next week, if you are signed up for next week, we will be, but nothing today.
Oh, okay. And will slides be shared, or is it just a lecture? Like,
oh, yes. Yes, I will share slides. I'm sorry. I had it on my mental to-do list to post slides, and I'm totally going to blame it on the virus because I've been, you know, working on at 20% since Wednesday. So yes, I will share slides. I will write myself a note. But yes, I will share these slides. And sorry, and if someone can yell and remind me, like when we take a break, I can probably even do it then. Good. The pen was on the. Wrong thing. Picked up my phone. Okay. Any other questions before I get going? Cool. Okay. Like I said, I'm doing background on high throughput sequencing. So I'm going to use the abbreviation HTS a fair bit. It's one. You'll also hear NGS, next generation sequencing. I kind of prefer HTS because this generation of sequencing has been around for like 20 years, so it doesn't really seem so next anymore. But those terms are used interchangeably. And I'm not going to really be talking anything about data analysis or software today. We'll be talking about that some next week for RNA-seq and in some future sessions for other applications. So speaking about applications, I'm just going to run really through. I'm guessing probably everyone here, if you're interested in high throughput sequencing, you probably are interested because you, in one way or another, are working with one or more applications of high throughput sequencing. So here are just like a few kind of like very high level examples of applications. DNA-seq, which isn't really quite an application, but it just means sequencing DNA. RNA-seq, which isn't exactly an application either, but usually when people say RNA-seq, they mean RNA sequencing for differential gene expression. Amplicon sequencing, which can also mean a lot of things, but I would say probably these days the most common application is for microbiome, 16S, targeted sequencing of bacterial ribosomal RNA, of the bacterial ribosomal RNA 16S gene. And there are many more. So I had just a few here: ChIP-seq, riboseq, Hi-C, methyl-C-seq. And just to like give you a feel, in case you didn't realize this, this is a poster. This is actually like kind of old now. I can't remember when I got it, but this is a poster that Illumina makes. If you contact the local Illumina rep, they'll probably give you one for free. And this is meant to be poster size. So if you can't read or interpret anything on here, that's because it's, I mean, I guess that's poster size, but the screens don't have like printed resolution. Anyway, so each row here is a different sort of specific application of DNA sequencing. And so you can see there's like, I don't know, I'm gonna guess at least 60 things here. And like I said, this is like kind of old at this point, like 2 or 3 at least years old. So like a ton of different ways that you can sequence DNA and use that information. So like just a few here. I'm gonna say are the most popular. I don't know if that's true. I haven't done a like careful survey. Oh crap, Richard, you're reminding me that I was supposed to include that in my slides and it didn't. I can see it now. Yeah, I'm monitoring you. So if you start chatting with people, then I'm going to know it. But you're reminding me that I was supposed to include that in the slides. I was also supposed to include the survey, which I didn't. Oh yeah. So should I show it now? Maybe I'll show it at the break for the people here. Okay. I'm not doing my job, sorry. Okay, so these are a few applications. So de novo genome sequencing. So for humans, like we have like a bunch of human genomes. So like that's not necessarily so interesting. But when there are organisms that are like newly discovered or like poorly studied, and you want a genome sequence of that organism, that's what de novo genome sequencing is. It's taking DNA from an organism that hasn't had its genome sequenced, sequencing it, and then like doing the downstream analysis to build out a genome and annotate it. Commonly used for both for research and in human health these days is different types of genotyping. So doing like whole genome association studies, trying to find, so GWAS is when you hear scientists have identified the gene for X, usually that means that that was identified through a GWAS study, genome-wide association study. But also there are other ways to identify genetic risk factors, whether like someone has a genetic risk factor that's been identified, or to identify novel genetic risk factors. And then identifying novel mutations. So this is pretty common. I mean, both genetic risk factors and mutation identification are used a fair bit in oncology to look at tumors and see like what kind of mutations those tumors have and potentially what they can be treated with. But also to identify, you know, did they inherit a risk factor that other members of their family might also have? And then therefore, should those people be, you know, subject to more careful screening? So this is the same poster I showed you before, but for RNA-seq. And again, you can see there's like a gazillion different applications for sequencing RNA, not just simple like gene expression. So here's just a few, you know, not all 60 of those. So sequencing the transcriptome, like it means, you know, basically sequencing all the genes that are expressed in a given sample. And you can do that for a bunch of different reasons, but I'd say the two most popular are, like I said before, differential gene expression. Also, if you've sequenced a new genome and you want to annotate it, one useful piece of information for that is what genes are actually expressed from this genome. Because there is software that can identify hypothetical genes in a genome, but it's not perfect. And having gene expression information can actually help help both eliminate false positives and false negatives. So that's a tool that's not uncommonly used for annotating genomes. You can also use RNA-seq for doing some of the things that you would do with DNA-seq, right? So if you have RNA-seq data, you can identify SNPs in the genes that in the the coding sequence of genes. It's not going to help you for the non-coding parts of the genome, but often what you care about is coding sequences. So you can identify SNPs from RNA-seq data. You can also identify gene fusions, which is when you get chromosomal translocations. You have sort of bits of chromosomes move around, and this is again something that happens a lot in tumors. And you can get two parts of the genome that aren't normally together, and that can happen in the middle of two different genes, and so you can get basically novel proteins that aren't encoded in normal genome being expressed in tumors, and those can be driving mutations for cancers. There's lots of known common cancers that are caused by these gene fusions, right? And so those can be identified from looking at the RNA from tumors. And just a tiny bit more specifically, when people talk about RNA-seq, there's sort of two major bins. One is bulk RNA-seq, where you take a collection of thousands to millions of cells, and you grind those cells up, you extract the RNA, and you sequence it. So in that case, you're getting an ensemble of gene expression from all those cells. You kind of get the average across all those cells, and that collection of cells might not be all the same cell type. That even if they are all the same cell type, they might not all behaving exactly identically because that's biology. And so you get this sort of summary of what's happening in that whole population. There's I'm going to say newer, although at this point it's not super new, technology for doing single cell RNA, or there's more than one technology for doing single cell RNA-seq, but this is a general approach. We're actually looking at individual cells and looking at the RNA that's being expressed in those individual cells. And then for amplicon sequencing, there are, again, also lots of different applications. Most often, amplicon sequencing is a subtype of DNA-seq, although you can do amplicon sequencing on RNA. And two applications that I see a lot is CRISPR barcode sequencing and, like I mentioned before, 16S ribosomal RNA sequencing for characterizing a microbial community, or I should say bacterial community. Questions about all that? That was like whirlwind tour, I know, but I just want to kind of give a little bit of context and motivation for what I'm going to be talking about for the rest of the afternoon. Okay, so let's keep going. This is a incomplete summary of different DNA sequencing technologies. And I think I grouped these by generation, but I think that most people will probably agree on them. So like technologies that have been around for like 50-ish years, like Max and Gilbert, Sanger, and Pyre sequencing. There are these newer technologies that have been around for like 10, 15 years. These are low throughput technologies. This and third generation technologies are what will be considered high throughput sequencing. And so there are a bunch of these. Some of them, I don't think this is on the market anymore. I'm pretty sure 454 is not on the market anymore. I'm not sure about Ion Torrent. Illumina definitely is. And then I have a special place in my heart for the third generation technologies. I'll talk a little bit at the end about those. But like I said, we're going to be focusing on Illumina. I'm going to come back to this in a minute. So this is like the 10,000 foot overview view of a high throughput sequencing experiment. I'm going to say that there are three major components. One is the sample collection, one is the nucleic acid extraction, and then the third one's the library preparation. The first two are going to be really very specific to the sort of system you're working in. And it's actually quite important that you pay careful attention to doing those correctly, because if you don't, then you can alter your results. You might not think that nucleic acid extraction is particularly important. I'll just tell you briefly that I worked on a project where we collected some samples, and these were human fecal samples, and it was a clinical samples, right? So we were enrolling people over like a 3 or 4 year period, and did a preliminary analysis on the samples that were already collected at like year 2 or something like that. And then once all the samples have been collected, like redid everything from the fecal samples and got fairly different results for the things that we'd already analyzed the first time around. And we puzzled over this for a while. And then we actually realized that the DNA extraction kit that we'd used the first time around, I think if I remember correctly, the company just changed it. I think it was the same kit nominally, but they changed it somewhat. And there were bacteria that were getting extracted with a new kit that were basically being missed in the first kit. Or if not missed, they weren't being efficiently extracted with the first kit. And so it shifted our results substantially. So that's just a little difference in DNA extraction. Yep. No, it's if I remember correctly, it was I can't remember which it was. I think it was gram-negative bacteria weren't being the gram-positive bacteria were kind of effectively extracted across both samples, but the gram-negative it kind of flipped. But I think it's the gram-negatives weren't being efficiently extracted with the first method, and the newer version of the kit, they'd done some things, I think, intentionally to improve that, but we didn't know that until we kind of went back and did a lot of tests. So nucleic acid extraction, important, but I'm not going to be talking about those first two things. Like I said, they're going to be very specific to the sort of system you're working in and the experimental question you have. And so what I am going to be focusing on, well, kind of really throughout today, is the library prep, because once you have the DNA or the RNA, things become much more sort of standardized. There still can be some details, like there's, again, you know, I showed you there are like 60 different types of, at least 60 different applications of DNA-seq. You know, it's going to depend on whether you're doing sort of amplify sequencing or shotgun sequencing. But if you're doing, say, shotgun DNA sequencing on, I don't know, I think that's a E. coli culture there, right? So if you're doing shotgun DNA sequencing on E. coli, it's going to be the same once you've done the DNA extraction as if you're doing shotgun DNA sequencing on human samples or platypus samples or whatever, plants, bacteria, doesn't matter. Okay, so like I said, we're kind of going to be focusing on library prep for the whole day, because thinking about the library prep helps understand like how the whole technology works at least for Illumina, I guess probably for all the technologies. So that's sort of what we're spending our whole day on. This is my 10,000 foot view of library prep. Just in case you're saying to yourself, well, what is a library prep? So a library prep is essentially you have purified nucleic acid and you need to get it into a format that's appropriate for sequencing. For many cases in Illumina, the Illumina sequencing context, what that means is that you're taking the DNA, you are usually making it into smaller pieces. So you can see here up there, that black line is a big piece of nucleic acid. Here we have smaller pieces of nucleic acid, and we have these different colored things stuck on the ends. Those are called the sequencing adapters. And so the whole goal of the library prep is to get the nucleic acid in a format that it's ready to stick on the machine and sequence. And so most of my goal for today is to explain mostly why we need to do this. I'll talk a little bit about how we do it, but the how is actually simpler than the why. Questions about this? Oops, okay. Okay, I'm gonna keep going. So to understand Illumina sequencing, I'm gonna start with Sanger sequencing. And there are kind of 1-2 reasons for this. So all of these that I'm showing you in orange, and I'll just remind you again that this is not all sequencing technologies. It's just kind of my favorites. I think they're probably the most popular ones. All of these in orange are all methods of sequencing that are sequencing variations of what you can call sequencing by synthesis. You shouldn't say that out loud too much because Illumina has a trademark on that term, but they're all sequencing by synthesis. And Sanger sequencing was the original sequencing by synthesis. And so you can, for the most part, understand all of these other methods with some variation, if you understand how Sanger sequencing works. I'll talk, so I'm gonna talk about Sanger, I'll talk about Illumina, and like I said, I'll touch on PacBio at the end. Okay. So I'm hoping that some people here have some biology background so that you can actually answer this question. So what are the minimum components? Do I want to try and use the whiteboard? Sure, I'll use the whiteboard. I don't know whether it's going to show up on. Zoom, but if it doesn't, it's okay. I have the answers later. The whiteboard's kind of for me to remember for the next 2 minutes. Okay, so if you want to replicate DNA in a test tube, what are the minimum components you need? That is totally correct, and yet I'm going to ask you to be a little bit more specific. Or explain why do you need DNA? Because we want to replicate that part. Okay, so what would you? Is there a specific term that you use to call this DNA? Yeah, there's a technical term I'm looking for, but yeah, template. Yeah, good. Yeah, so the template that you're going to replicate. Polymerase. Yeah. DNTPs. Yep. DNTPs will be, it's fancier to say DNTPs. I like that better. I did my PhD. There's a postdoc in the lab where I got my PhD who was a nucleic acid chemist. So he always like beat me up over like using primers. I think that's it. Let me check my answers. Yeah, good. Okay. So you got the all the key things. And I have a star here, and I think I put that star there because often people use thermotolerant DNA polymerase, but you don't actually need a thermotolerant DNA polymerase. It just makes life a little easier. Okay, so these are the basic ingredients. I'm just going to go through, in case you don't know or in case you don't remember, how the DNA synthesis works. Do I want to use this? Yeah, I'll use the, oh no, it doesn't show up. Oh no, there it goes. Now I should get one of those like fancy pointers that you can actually see really well. I don't know how to do that. I'll just do this. Okay, so here's the basics of DNA synthesis. So you have this gray blob here that's the DNA polymerase. This here, this longer strand of DNA is the template. Here's one nucleoside triphosphate. And let's see, we had polymerase, template, nucleoside triphosphate, primer, that was the other thing. Yeah, and so you can think of this as the primer. I mean, it is a primer, right? And so primer is just a small piece of DNA, but DNA polymerases can't synthesize DNA out of nothing. They actually have to, or not even out of nothing. They can't start on a template without, they need to attach the next nucleotide to something existing. So they need a little bit of nucleic acid to attach the next nucleotide to, and that is the primer, right? So this is sort of like a little bit of DNA that's complementary to the template, and that the next base can be added to. And so the synthesis is the polymerase is here, a nucleoside triphosphate that is complementary to the next base on the template, just randomly is kind of like diffusing around. It happens to land, in the active site of the polymerase. The polymerase recognizes, well, this base is complementary to the base in the template. So we're gonna go with that one. And so it breaks this nucleoside, this triphosphate here. It releases this dimer of phosphates, which is called pyrophosphate. You don't need to know that, but there's a bonus fact for you. And then this phosphate that was closest to the new base gets added here. It gets linked to the previous base, and it forms the phosphate backbone of the growing chain, right? So now you just added one base. Everyone good with that? Questions? Sounds familiar? Okay. So you needed to know that because Sanger sequencing is a slight variation on that. I don't know if this tea is working anymore. I might have to go for the cough drop. Okay, so Sanger sequencing is like I said, a slight variation on that. So we're going to focus on what are we focusing on? We're going to focus on this test tube here that's green and is labeled C, and I'll explain in a minute why it's labeled C. So here's our template. It's a little tricky to follow because it curves around like a double helix. Here's our primer. The polymerase isn't here, but you can just imagine it. And so we're doing Sanger sequencing. So it's just like what we did before. You got your template, primer, polymerase, you got your DNTPs, and you got something extra, which is this right here, which is a C analog, and it's what's called a terminator base, because it can be added to the growing chain, but then once it gets added, another base can't be added after it, right? It's kind of like a stop sign. And so if it gets incorporated, then that's the end of the game for that, for replicating on that template. And so you can imagine here, this is a template, and so it's going along, and the reason why this is green and is labeled C is because it's got a C terminator in it. It's got all the normal bases plus a C terminator. And that's important. You have to have all the normal bases for this to work. So, you know, in this tube, what's going to happen on this specific template is that the polymerase is going to come along. It's going to add a T here. It's going to add an A here. It's going to add an A here. It's going to add a G here. It's going to add a T here. And then here, this G, there's a probability that it will add a normal C, and there's a probability that it'll add the terminator C. And there's one thing that you need to keep in mind, which is in this test tube, there's not just one template and one primer and, you know, a handful of nucleoside triphosphates and a handful of terminator Cs. There are billions of template molecules, there are billions of primers, or probably maybe trillions, and probably similar order of magnitude trillions of molecules of the DNTPs, right? So because you have so many things there, on some fraction of the templates, you're going to get a normal C incorporated here, and on some fraction, you're going to get a terminator C incorporated. That makes sense to everyone? Questions about that? Okay. And so if you had the normal C incorporated here, then the polymerase is going to keep going and add a G here and a T here. And then again, at the next G, you have this probability that you have a normal C versus a terminator C. And then the same for that, for this next G. And so what you end up with is, in this test tube, you end up with a mixed population of molecules where some of them got terminated at the first G in the template. So you get this. Some of them got terminated at the second G in the template, so you end up with this. Some of them got terminated at the third G in the template, so you end up with this. Everyone okay with that? Okay. And so we have, so I told you, well, I don't know if I told you or not, but like we started with the C tube. So we also have an A tube, a T tube, and a G tube. And so the same thing is going on in those. You can imagine that in the A tube, you have everything that was in the C tube, except instead of a C terminator, you have an A terminator. T tube, you have a T terminator, and the G tube, you have a G terminator. And so each of those reactions are happening separately in those tubes. And then once they're, you know, you've let it run long enough, you take it and you run it out on a gel, which, just in case you don't know, is just a mechanism for separating molecules based on size. And so here, these molecules are all going to be different size. You can imagine that if you look across all the tubes, you're going to have a bunch of different molecules, different size. And I always get confused about which way to read this. Okay, we want to look, if we're talking about C, we want to look at this C lane. And so what you can see is that there are three different bands. And these two bands are right next to each other. So these represent this band here represents this smallest molecule. And then these two bands represent this one and this one. And so you can look at this gel, and the smallest molecules are here, And so you can look at the very smallest band and you can just walk up the gel, starting from the smallest band to the next smallest band, to the next smallest band, to the next smallest band, or I guess the next largest band, whichever you want to call it. And you can read the sequence. And so if you want to actually figure out what the sequence of the template is, you got to switch it, right? So here, the smallest thing is a T. So that tells you that there was an A on the template. Oops. And so here it's got the template sequence along here, right? So A on the sequence, on the template, T, C, then we go back to A, and then we go to G, right? And so this is the first one that's from our C tube. Does that make sense? And so you can just like walk up the whole gel. Everyone got that? How you read this? Okay. And so like, just because it's super fun, at least for me, okay, it's time for a cough drop. Excuse me. Okay. So this is the figure from the original paper where this method was published. The first author is Fred Sanger. That's why it's called Sanger sequencing. It was actually done in his lab. So you can like read up this gel. Fun fact, back in the old days of Sanger sequencing, after you did the sequencing, you got to sit there with a big gel and read the sequence off, which you can imagine was a little bit error prone. I was fortunately like just got into research around the time when this was being automated, which I'll talk about in a minute. So I never had to read one of these gels. Little aside, so Fred Sanger got a Nobel Prize for this. I'm trying to remember the order. I think this was his second Nobel Prize. I think it was previously that he got Nobel Prize for developing the technology to sequence proteins. He worked for a government lab in England, and so they had a mandatory retirement age of, I think it was 65. So he retired at 65 after getting his 2 Nobel Prizes, and then spent the next like 35 years of his life puttering around in his garden, apparently. Any day when I think that I've like, had major accomplishments, I try to remind myself that Fred Sanger got 2 Nobel Prizes by the time he was 65, and a little bit of reality check. Anyway, back to sequencing. So like I said, by the time I was getting into research, this process had been automated. And so the automated sequencing, it's called dye terminator sequencing. It's almost exactly the same as what I just showed you. Sorry, were there any questions about this before I keep going? Okay. So it's almost exactly what I just showed you. There are like 2 technological advances here. One is that instead of just the sort of basic base terminator that I mentioned before, this is called dye terminator sequencing because they've incorporated those terminator bases that stop the sequencing, that like, or block the chain. They have a fluorescent dye chemically attached to them. So that's number one. And number two is that because now the fragments are fluorescent, you can basically detect them with a laser and a light detector. Okay, so the laser is set at the right frequency to activate the fluorescent dye, and then the dye fluoresces, and you can detect what color light is coming off. So instead of having 4 lanes here, and you have to like walk up the dye manually, walk up the gel manually, you only have 1 tube, and because you can distinguish here, the way we distinguish like what base was incorporated was by which tube it was in. But here, because A is a red sorry, T is red, A is purple, G is green, and C is blue. We can distinguish what terminator base was incorporated just based on the color. So we don't need to separate it into tubes. And so we just do the reaction in a tube, put it on this gel. We have the laser sitting there, and as the bands run past, I don't think I mentioned this before, but you use an electric charge across from one end of the gel to the other. So this is what it looks like when it's done, but everything actually got loaded here on this end, and then the smaller things move faster than the larger things. So the smallest things the smallest fragments end up farthest on the gel. Same thing here, you load it up here and you run it. And so the bands just sort of run past the laser and the detector. And so hooked up to a computer, and the computer just reports red, purple, blue, red, green. And then okay, well, there was a T and then an A. Keep on hitting that. T and then an A and then the C and then a T and a G, right? And so you just leave that, you come back later and the computer has the sequence. Big improvement. But only kind of sort of little tweaks in the technology. Makes sense to everyone? Questions? Well, it depends on what, so it depends on what you report. But so I think that, so the terminator is, the red is a T and the purple is an A. So so if you wanted to get the template, so here it's reporting what's coming off the gel. But so if you wanted to know what the template was, then you'd reverse it. Or you complement it. And I guess complement. Just complement it. Wait. Yes, just complement. Okay.
What are the height rates again?
So I'll show you here. So what the data really looks like is you can think of it as 4 different graphs that are laid on top of each other, where this axis, normally these are sideways, but I'll explain why this is like this in a second, or maybe you can figure out. So this axis is time, and this axis is intensity of the signal of that color. And so, like I said, this is what the data really looks like. So you can see the intensity of this red signal here is a little bit higher than this red signal here. Usually, if you have a nice data set, it doesn't really matter that much. I will talk in a few minutes about where it does matter. But this is what the gel would look like for dye sequencing. This is what the data that you get looks like. And then this is that same data if you did traditional Sanger sequencing, because you can kind of see it side by side. Other questions? Okay, so that's the background we need to start thinking about high throughput sequencing. Let's talk about high throughput sequencing. So again, this ends up being a little bit of a kind of incremental step from that dye terminator Sanger sequencing that's going to give us a lot more data. But we're going to need to get to some other tweaks in a little bit. Okay, so here's our DNA template, and you'll recognize these adapter sequences that I mentioned before, that the whole goal of our library prep is to get those adapter sequences stuck on the nucleic acid. So in this case, this is what we're interested in. I'm showing you the sequence for demonstration purposes, but in general, this is a black box. We don't know what the sequence is. We do know what the sequence of the adapters is because we've engineered those specifically because we need them to be certain sequences. And I'm going to explain why we need that in a little bit. right now I'm going to show you one reason, which is that we're going to use this as a priming sequence for our sequencing reaction, right? So back here, when we had a specific template we wanted to sequence, we needed to actually design our primer to match that template, like right up to where we wanted to sequence it. Here, we don't need to know our template sequence because we've stuck on this adapter, and we can use part of the adapter sequence as a complementary sequence to design our primer, right? And so here we design a primer that's just complementary to this part of the adapter. So that makes things easy. Okay, so now let's talk about how this is really Illumina sequencing. It's going to be different on some of the different platforms, but sometimes different, sometimes similar. We'll get to those details in a little bit. Okay. So we have our template with the adapters on, and the first thing that we're going to do is we're going to add primer. So it's going to hybridize. Oh, did I take out the thing about hybridization? Maybe I show that later. Okay, just for now, we'll, this primer is it's gonna interact with the adapter in a sequence specific manner. Okay, so the next thing we're gonna add into this reaction is our polymerase and our di-terminated nucleoside triphosphates. Now, something that I forgot to mention before, but maybe it's obvious, is that here we also have our normal DNTPs and our di-terminated DNTPs. It's a mix, just like here. Here, we only have di-terminated DNTPs, okay? And so the polymerase is gonna bind to the primer and the primer that's hybridized to the adapter, and it's going to say, okay, well, it's not saying anything, I'm anthropomorphizing, but it's going to do its job, which is to recognize when the appropriate nucleotide has entered the active site, and it's next to where the base needs to be added on the complementary to the template, and so it's binding there, a T is going to end up landing there, and it's going to end up getting incorporated. And so now we have a T there, and it's terminated, is di-terminated, so nothing else is going to happen. Sorry, and the thing that I should have said is that once it gets added, you incubate it for some time, and then you want to get rid of all the excess stuff. So you want to get rid of the polymerase, you want to get rid of all the excess di-terminated GNTPs. So it's going to wash that away. We'll get into details about how that happens in a minute. So now we have our template with a T incorporated. And so now we need to do some imaging, just like we saw on the di-terminator Sanger sequencing, to figure out what base was incorporated. So we're going to zap that with a laser and then capture the image. And so now we know, okay, it's red, so we know that a T was incorporated. So we've sequenced. But we're not done because we only sequenced one base. So the next thing we're going to do is we're going to get rid of that di-terminator, because what I didn't tell you before is that this is a reversible terminator. You can flow in the right chemical, it'll take off the di-terminator, and then that T is ready for another base to be added. So we're going to flow in the chemical into this reaction, or into like where this template's happening. Now we have a T ready for a base to be added. We throw in some more polymerase and some more di-terminated nucleotide triphosphates. And polymerase is going to bind again. The complementary nucleoside triphosphate is going to It's going to like get in the binding pocket. The polymerase is going to, cleave the pyrophosphate off and use that, the last phosphate as the phosphate backbone. And so now we're going to wash out all that excess stuff, and we have a C with a C dye terminator on it, stuck on after the T. We image that again, and now we know it's blue, so we know that there was a C incorporated. Makes sense to everyone?
The only thing you remove is also a tradition of a terminated dye is the dye.
The dye and the dye itself isn't the terminator. It's the terminator is like the dye is attached. So the terminator, yeah. So you're taking off the terminator, and with it, the dye comes. But everything else now, it's just once we've removed that terminator, now you just have a normal C there. And so now,
Where is the normal C coming from?
because you had a dye terminated C, and the way that this is getting into chemistry, which I'm not a chemistry expert, but basically the way that the dye terminator was designed, you can chemically treat it to actually take off the terminator, and that leaves you with so like when you have these terminated bases, it's the normal base, but with something stuck on the end, basically, bit stuck on the 3' end. This actually like the 3' carbon. I don't know what, maybe it's nitrogen. I'm not sure what's actually there normally to terminate it, but it's in this case it's chemically removable, right? And so you end up with just a normal C there that you can now add another base to. And so we can, kind of keep doing this. We put in more polymerase, put in more nucleoside phosphates. So now G is going to get incorporated, get rid of the excess stuff, image it. Now we know that it's a G. That makes sense to everyone? Okay. So there are some complications here that I've sort of glossed over. So one I'll explain right now, which is, this is what I showed you before. It's like a standard chromatograph from Sanger sequencing. And we talked about before how the peaks are a reflection of the height of the peak is a reflection of the signal, right? And like if I gave this to you and I didn't give you this top part here, like I could explain to you in like 30 seconds how to read this, and you guys could like write out the sequence. It's pretty easy. Like there's software to do this, but it's pretty straightforward, right? You can tell that that's T, that's an A, that's a G, right? So this is good data. This is bad Sanger data. You see that almost every one of these peaks, it's there's actually like 2 signals there. So this happens either if you accidentally put 2 separate templates in the tube, or if the primer recognizes multiple sites in the template. And so you end up with, at each position, a double signal. There are a few places where there's a single signal, and that's where the 2 different templates happen to agree, right? So like in both places, whatever this is, like 11 after the beginning, there's an A there. Great, I know there's an A there. And you might say to me, well, Josh, it's not so bad, right? Because I can see here that this is an A and a G, right? That's not too bad. And here I can see that it's a G and a C. So that's okay, right? I got some information here. Well, the problem is that that's true, but you don't know, does this A go with the C here, or does it go with the G here? So you know the average, you know, like kind of the, across those two templates, you know at each position that it's either, you know, A or C, or you know that it's, you know, T or A or whatever, but you don't know which goes with which, right? So you kind of have like statistical average at each position of the base, which is better than nothing, but it's not really that great. For example, if you gave me a DNA sample, I could tell you that at each position, it's either A, T, G, or C. You know, I could give you back like millions of bases of saying, well, the first position is either A, T, G, or C. The second position is either A, T, G, or C. The third position is either A, T, G, or C. And what do you know? You don't know anything, right? You have no additional information from what I gave you, or you know what you initially gave me. So this is kind of useless. Now imagine when we're doing high throughput sequencing, we're sequencing at this point billions of fragments at a time. So here's two different things being sequenced at the same time, and we have it mixed up. So if you have billions of signals at the same time, you're just going to get this sort of like average of the position. If you can't separate those signals, you're just going to get the average of those positions, which is, like I said, it's A, T, G, or C at the first position, A, T, G, or C at the second position, A, T, G, or C. And basically what you end up with is the GC content of, or the ATGC content of across the whole genome. Well, I guess it ends up being the GC content across the whole genome. Like that's what the height of those peaks are going to be. So this is like kind of useless. So one of our challenges I just showed you kind of in part how we can do high throughput sequencing, but one of our challenges is we need to be able to detect the signal from individual molecules, or at least individual fragments. And so we need to be able to sort of like separate those fragments. We need to be able to, you know, because we're sequencing, like I said, billions of fragments in parallel, you know, simultaneously. You need to be able to somehow separate those signals, be able to detect those separate signals. And the other technological advance that we need is that removable terminator, which you already saw, right? Okay, so that separation bit, that's been solved a couple of different ways. This is from an old article that shows how that's done for different technologies, several of which no longer exist or are no longer on the market. There's Illumina. I think Illumina, yeah, Illumina's here. I can summarize all of these for you simply, which is you stick the fragments you want to sequence on some sort of surface, and you kind of spread it out on the surface. So like imagine I like had a container, maybe I should do this sometime. I had a container full of jelly beans here, and I like threw them out. Like maybe I like had them in like, if I had them in a jar right here, it'd be hard to tell like what the different colors are. But if I like threw them out there on the ground and I made a big mess, I'd have a big mess, but also it'd be easier to see like each of the different jelly beans, right? And so that's kind of what you're doing here. You're taking the fragments and you're throwing them out. You're throwing them out and attaching them to a surface. And then it makes it easier to like detect the signal from each of those fragments. So for Illumina, what you end up doing is you end up having a flow cell, which now, for those of you who are here, you're in luck because you get to see what I'll hold it up for those who aren't here, just so you can get an idea of the scale. Here's one flow cell, that's NextSeq 500 flow cell, and here's a MySeq flow cell. Actually, I will pass these around, and then I've been talking for an hour, so maybe we'll take a break. And actually, let me tell you what you should look at. So here, if you're online, you can look at this, which is, so each of these, there are 4 things on this flow cell. These are each what are called lanes, and at the end, it comes to a point, and that's because like on the underside, there's a little port, a fluidics port, so that liquids can be flowed onto that lane, and then actually at the other end, it comes to a point so that liquids can also be taken off on that. So it's flowed from one end to the other. What else do I want to tell you? So basically, this is a fancy piece of a fancy microscope slide. Like all this stuff here is just plastic. It's like kind of infrastructure. Actually, sorry, I should say it's two microscope slides stuck together with like these channels carved out in these channels here that are carved out. On the MySeq flow cell, so here there are 4 lanes. On the MySeq flow cell, it's actually just one lane that's in a U shape. And so the fluidics, like here's the input port and here's the output port, or I might have that reversed, I'm not sure. But yeah, so basically it's just like 2 glass slides stuck together with like a channel carved out. And then like there's some rubber seals on the fluidics, on the port for flowing in, flowing out. Okay. Questions before we take like maybe let's say 10-minute break. So I have 2:05 now, so maybe we'll come back at 2:15. Questions? Okay. Here, actually, for those of you who are on Zoom, I'll just leave those pictures up so you can see them. But I realized that I didn't exactly answer the question about how much they cost correctly, because these were free because they're used. Yeah, so I'm not like, yeah. So once these are basically single use. Once they've been used once, I don't know if now they recycle them. I know that Illumina, for some things, they've tried to like reduce waste and stuff. But these I got from the sequencing core several years ago, once they were done, because they were just gonna throw them out. And I think it's nice to actually like see them in person. Okay, any other questions that have like popped into your head since before? And I'm gonna say
cutoff for peak height when you're looking at when you're QCing like sequencing data, do you like cut off, because when you showed one of the peaks on what there was a lot of overlap. I saw some that were really tiny, like 3 of them on the wall, like in the same.
like here.
Yeah.
Yeah, so
Are those still like?
so when you see real data, this is like super nice data. And what you should keep in mind is that, like I said before, this is 4 graphs that are overlaid over each other. And so there's always like, there's no like perfect signal. So there is going to be like a little bit of signal in the other channels. Almost always when you have a Sanger data, if you look carefully, you can see like little tiny peaks. But that's just, it's either noise in the detector or it's misincorporation of bases, because the polymerases, specifically the polymerase that's used for this, is fairly error prone. But you can tell, right? I mean, like this, it's pretty easy to see, like that's a C, right? And even if there's like a little bit of like an A peak or like whatever, you could tell. So for Sanger sequencing, there is software that not only processes this and gives base calls, but it also actually gives you a confidence information. So it's called a quality score or Fred score. And that's a prediction of the accuracy of the call. And so for Sanger sequencing, you tend to get Fred scores in the range of like 60, which is basically an expected error rate of 1 in 10 to the minus 6, so like 1 in a million error. We'll talk a little bit more for Illumina sequencing. The error rates are more like 40, which is 1 in 10,000, 1 in 10 to the minus 4, which is obviously a lot higher. And those numbers are like, the difference of those numbers is much more important because often with Sanger sequencing, you're, the most that you can get out of a single sequencing read is about 1,000 base pairs. So if you have 1 in a million error rate across 1,000 base pairs, it's likely that those are all actually correct. For Illumina sequencing, if you have 1 in 10,000 error rate and you're generating 10 billion base pairs of data, there's a lot of error in that data, right? It's, you know, whatever 10 billion divided by 10,000, right? So that's a million. Yeah, so that means like out of 10 billion, and sorry, it's 10 billion. No, yeah, I guess it's 10 billion bases that you get from like the newer machines off a flow cell. Actually more on some of the flow cells, but right. So like a million of those bases are wrong, or like you're assuming that they're wrong. And that's only, that's at like the sort of best quality you can get off Illumina. And not every base is actually 1 in 10,000. Some of them are lower quality. Well, you get that there, right? So in this case, yeah, you get like very low quality. I mean, well, you can see that the calls are actually, here are the base calls at the top, and N means that it doesn't know what to call it, right? So like C is, I mean, it'll also give you an, you know, predict the accuracy, but the accuracy is going to be like, you know, basically, you know, 1 in 4. I mean, I don't know what it'll give you for that exactly. Other questions? Okay. Oops. Okay, so let's talk a little bit more about kind of like giving you the high level details, and some of what I've already told you is somewhat Illumina specific. I'm going to dive into the Illumina pool a little bit more. And like I said, so SBS, sequencing by synthesis, is actually, I believe, trademarked by Illumina, even though like trademarked, not patented, but that term, if I remember correctly, is trademarked by Illumina. And I should say that there are a couple of reasons for kind of focusing on Illumina. I'd say one of them is that the vast majority of sequence data is generated on Illumina sequencers. They have, at least in this country, no, I mean, across the world, except for some private companies that have developed their own in-house sequencer, or I can think of one private company that's done that. The vast majority of sequencing is done on Illumina sequencers. But like I already said, it also, like understanding how Illumina works, I think it's not too hard to understand like other technologies. And I'll try and take a few minutes at the end to tell you about this. Okay. So as you know, this is a real flow cell. So here is my like theoretical version of a flow cell. It's just a rectangle, but that's going to work for us. So the first step, and I don't know if I said this or not, but it's called a flow cell because you can flow the reagents, the sample and the reagents in and out. If you look carefully at the back here, there's these little like rubber things that cover up the like pointy bits at the end where the reagents get flowed in and out. So the first step, once the library's been prepared, and we haven't actually gotten to library prep yet, but once the library's been prepared, the first step is actually loading the library onto the flow cell. And so that's just flowing it. You know, the machine does this, but it just basically gets pumped onto the lane. And so, and in the, basically it just, the fragments in the library are just gonna flow across and they're basically gonna randomly stick in different places across the flow cell. Kind of like when I mentioned like throwing the jelly beans on the ground, right? You can get like a random distribution of jelly beans. So we're gonna keep it simple here and just talk about four fragments, because if we tried to do 10 billion, it'd get out of hand. Or not 10 billion, like a billion, it'd get out of hand. So for our library, we have 4 fragments that are stuck on the flow cell. And so now let's do some sequencing. And this should look familiar. The next thing that we're gonna flow onto our flow cell is the polymerase and the di-terminated DNTPs. And so polymerase is gonna bind, it's gonna incorporate a base. And so now you're gonna have these fluorescent dyes attached to each of those fragments. You're gonna get rid of the excess and you're gonna image. And then, so in this case, like I said, this is like basically like a fancy microscope slide. And so the imaging is actually in the sequencer, there's built in a confocal microscope essentially. And so it's just gonna like take a picture, illuminate with a laser, take a picture. And now you're gonna know at this position, you have a G, and the colors are kind of funny. I swear this is actually a different color. I think it's green, maybe. Oh, okay. Oh, yeah. Wow, that's a huge difference. Okay. Actually, on this screen, you can kind of see that it's green on my screen. Better on that one. Anyway, so you can tell at each position, right? So this is random fragments that have landed randomly on the flow cell. And what you're going to know is at this position, you have a G, at this position, you have an A, at this position, you have a G, this position, you have a C, right? And now you're going to reverse the terminator. So now there's no more fluorescence. And, but what you record is in the sequencing, what the computer is going to record, or really, I mean, it's taking the image and then the image is getting processed offline. But the way the image is processed is the XY coordinates of this position. You're going to write down, okay, well, at what is this, like, XY coordinates of like 3, 4, you're going to write down, okay, well, I saw a G there. And then the XY coordinates of like 10, 5, you're going to write down, okay, there's an A there. That makes sense? So if you're kind of taking notes, that's what's going to be recorded, is the base that was observed and the position that was observed. So now you do you get rid of the dye in the terminator. You're going to do another cycle, flow in the polymerase and the DNTPs. The base gets incorporated, get rid of the excess, and image it again. And so the information that you're going to get is the position and the base. And we just happen to have a record, so we're not going to see this G again. We've recorded it in our notes that at that position we saw a G before, right? And here we saw an A before, and we happen to be seeing an A again. Does that make sense? Right? And so, now you're bored with this, right? We get rid of the di-terminator, we put in some new polymerase and fluorescently terminated DNTPs, get incorporated, do an imaging step. And so now we know at each of those positions, okay, we have an A there, we have a C there, we have a G there, we have a C there, right? So at the end of 50 cycles, when we compare our notes for each position, what we have is a 50 base pair sequence from that position. And we don't care about the position is just random. That's just for record keeping so that we know for each base, we know which previous 49 bases it went with, right? Does that make sense? So ultimately, what we're doing is we're doing these cycles of sequencing. And so when you hear about, if you've ever ordered Illumina sequencing, or if you've ever worked with Illumina data, you know, you might hear about 50 base pair reads or 100 base pair reads. And the number of bases that you're sequencing, the number of bases that you're getting reads is just the number of cycles of that. Putting in the polymerase and the terminated diterminated DNTPs, letting it get incorporated, imaging it, reversing the terminator, and then doing that over again. So if you have 100 base pair reads, then you did 100 cycles. So for Illumina, the size range, the sort of normal range of reads that you can get is between 50 and 300. You might be able to go below 50 if you like request it special. You pretty much can't go above 300. It's sort of like a limit of the technology. I mean, in theory, maybe you could, I don't know. I've never actually worked directly with machines, so maybe you can get it to give you more cycles, but the quality actually tends to drop off with the longer reads.
What is the typical length of the fragment?
So that's a good question. The length of the fragments is actually usually a distribution of sizes. Actually, can I answer that in a few minutes? Because I'm going to get to it, like maybe in, I think, I'm going to say within 5 slides. I can't remember for sure. Other questions? Okay. I think I'm going to answer it. If I don't, remind me. Okay, so here are like a few details of things I glossed over. So I'm answering it now. So the fragment size, this piece of DNA, this black line, which is representing a piece of DNA, for input to doing library prep, usually you're looking at 200 to 1,000 base pairs. It can be smaller, but that's sort of what you're aiming for. It can be a little bit bigger, but not really a lot bigger. And so this is like a slightly, again, like a little bit of a sketch of the library prep. So again, you have this fragment that's DNA fragment that's about 200 to 1,000 base pairs. You're sticking on adapters to either end. I haven't told you too much about the adapters yet, except for the fact that you're priming off the adapters for sequencing, and you end up with with this bigger piece of DNA that's what you're interested in, plus the adapters, and that's your sequencing library. Now here, we're just looking at one fragment, but the sequencing library is a collection of like billions to trillions of fragments. Here's where I talk about hybridization. Maybe I should have before. So in case you're not familiar with hybridization, I'll review this for 30 seconds. Basically, if you have 2 pieces of DNA and their sequences are complementary, they will interact, but they'll have a relatively weak interaction, base pairing interaction, that is sequence specific. So G's pair with C's and T's pair with A's. You probably all know this, but just in case you don't. So that interaction is relatively weak, but when you extend it over, say, couple hundred base pairs, or like a whole genome, it actually ends up being pretty strong. And so like to separate big chunks of DNA that have hybridized, well, the easiest way to do it is actually you can do it chemically, or you can do it with heat. So like you get the DNA close to boiling point and they'll separate. You also like put it in the right combination of salts. You can also get them to separate. So that's hybridization. Questions?
Single strand?
Yeah, so these are single strands of DNA. Right. And I will say that in cells, living cells, most of the DNA is double-stranded at any given time. So like this, and also you tend to have like whole, right here I'm just showing like a little fragment that's hybridizing to this bigger fragment. That's not something you normally see like in a normal cell. Okay, so that's one little piece of background that I want to give you. Another piece of background that I want to give you is I'm going to walk really quickly through PCR. I'm assuming that people have seen this before, but just like a little bit of a reminder, which is that much like we talked about the in vitro DNA synthesis, one way to do that is a technique called PCR, polymerase chain reaction, where you can make billions of copies of a fragment of DNA. So before we talked about a primer, here so this is the bit, or I guess here is the bit of DNA that you want to make lots of copies of. So here's double-stranded. You heat that up to about 95 degrees Celsius. That'll unwind the DNA. So now it's single-stranded. If you cool it down a little bit, the primers can bind to it. And so you're gonna have, you're doing PCR right, you're gonna have one primer that's gonna match this strand somewhere around here, one primer that'll match the complementary strand somewhere around here. So they're gonna be pointing towards each other. And so then if you add in polymerase, you get this blue bit, right, that's newly synthesized. So now you had double-stranded DNA, you denatured it so it was single-stranded, then you add in polymerase and DNTPs, you allowed the primers to bind, to hybridize, and now you have double-stranded DNA for at least part of it, right? So this direction is still single-stranded, this direction on the complementary strand is still single-stranded, but here you have double-stranded and here you have double-stranded. So now you have two copies of this region. If you just do that cycle again, now you're gonna have 4 copies of that region. And what ends up happening by the time you get to, like, say, you know, if you do that cycle 20 or 30 times, the templates it's gonna end up being that, you know, this end is defined by one of the primers and this end is defined by the other primer. So you're gonna basically just be amplifying on top of previous copies that you made, right? So you end up, for this region here, or this region here that you design the primers to, you end up with millions, billions of copies, whereas the rest of the genome, the rest of the DNA that was input, you're gonna just have one copy of all. Does that make sense? Questions about that? Okay. So all of that is background because I need to tell you that I wasn't entirely honest with you before. I glossed over some details. So one of the details I glossed over is that the Illumina sequencing method is not sensitive enough, that confocal microscope is not sensitive enough to detect single molecules of the fluorescent dyes. It really needs like millions of molecules of fluorescent dyes. And so we essentially have to amplify that fragment that we're going to sequence so that we can get enough signal to detect it. And so the way that works, actually I glossed over another thing first, which is that I just kind of like waved my hand about the, you know, when I said you flow the library and it just sticks. So I kind of waved my hand about that. But the way that it sticks is that the flow cell, not this one, because this actually has fragments of DNA that have been sequenced on it now. But when you get a new flow cell, attached to the glass are little pieces of DNA that are complementary to those adapters the orange and green adapters that I told you need for sequencing that get put on during the library prep. right, that are necessary for sequencing. And this is one of the reasons why they're necessary, because when you flow the library onto the flow cell, those fragments are going to hybridize to the short pieces, the oligonucleotides, the short pieces of DNA that are chemically attached to the glass, and they're going to stick to those, the ends, the adapters are going to stick to the DNA that's on the glass, right? So it's not just like magically sticking, it's actually hybridizing to the little oligonucleotides that are on the glass. Everyone okay with that? Questions? Okay. So the next thing that I glossed over is that after you hybridize the DNA, there's an initial step where so this is single-stranded. This oligonucleotide can essentially act like a primer. So if you throw in some polymerase, now you can make double-stranded DNA, right? Does that make sense? And then what you do is you denature this, so you get rid of the initial fragment that like was flowed in, you get rid of it. And so now what you have is a piece of DNA that is it's covalently attached to the glass slide because it was extended from this oligonucleotide. Basically did like one cycle of, you synthesized, you did in vitro synthesis on this surface and made a new piece of DNA. Got it? Questions? Make sense? Confusion?
Doesn't that happen during the bridge?
We haven't gotten to that yet. This is before the bridge amplification. This is like the precursor to the bridge amplification.
How does it become before the bridge?
It's a good question. So I didn't design this, but I'm pretty sure the reason is that this piece of DNA is just stuck on the flow cell by hybridization, like just at this is basically 20 base pair hybridization. The bridge amplification, you're going to do these cycles of amplification denaturation. I mean, bridge amplification is basically PCR, like stuck on a surface, right? So I assume that the reason why they decided to do it this way, well, actually, I'll give you two answers. One, I think the reason why they decided to do it this way is because now you have it covalently bound, right? The other reason is that now you're actually, the sequence that you're getting off is actually the sequence of the template. It's not a big deal, right? You could reverse complement it bioinformatically, but it is, right? So you're actually like, the template that you're using is, or the sequence that you're going to be getting off is actually the direct sequence of this. It doesn't actually matter for most applications. And you could, again, you could do it bioinformatically. Yep.
How do you ensure that you have like unique fragments in every location, at every core?
Well, so it's, the answer is number one, you don't know. You actually might get the same fragment in multiple places. That's okay. Because you've just basically randomly, so usually you've like randomly shredded the genome. And so the fragments that you're getting it's just a random sampling. The other answer is that the location, which I'll get to in a second, I'm gonna tell you another lie and I'll tell you the truth later. Like I said, it's like the jelly beans where you throw it on the ground. So this is actually an image of what a flow cell looks like. And each of these spots are where you had a fragment land. And so you can kind of see it's pretty random, right? That answers your question mostly for now. Do you have other questions? I'll tell you the truth later, but if you didn't know that I lied to it a little, I mean, this is the truth. It's just not the truth. This is the truth for this. The technology's changed a little bit, so like the newer sequencers aren't quite like this, but it's close enough. I can tell you the details of that later. Okay, so here's where the bridge amplification comes in. And this is, like I said, it's basically just a PCR. because you have all of these oligonucleotides that can act like primers here. And so you do this PCR cycling, except instead of having free, having the primers in solution, all the primers are on the flow cell. And so you do, this slide is old. I don't know if you're still doing 35 cycles, but that probably makes sense. So you start with this one fragment that was bound here, randomly bound there. You do 35 cycles and you end up with millions of fragments of DNA that are exact copies stuck in this one spot. And so this is showing you not that one fragment, but just like detecting the DNA, and you can see that you just have these like spots that are randomly distributed across the flow cell.
Isn't like within that one spot, you would have like the reverse of both of them giving signal in a single spot?
Yes and no, because remember when we're doing the sequencing, we're priming off. So we specify which strands. This guy here, you are. Yeah, it's actually a good question. I assume that this one you actually prime here, and this one you're priming here. I don't know if there's anything that would block that, but yeah, it should work out okay, right? Because you're priming off one end.
Because you have to go, regardless, you have to go both. You have to go, either direction depending on, where they are. I guess like when looking at it from the flow cell, but from the primer that you're sequencing, you're going the same direction.
Yeah, and the imaging is not, so I'm pretty sure that's actually, that limit of like 1,000 base pairs, I think part of the reason is actually just the imaging, right? If you get much longer than that, then you start to worry about like that's a guess on my part, but I'm assuming that's one reason. I could be wrong.
I don't think you could do the same primer going both ways. You could have 5' 3' works with those two strands, but I can't quite.
No, it is, right? Because this is the, oh yeah, sorry, you're right, because the primer might be going that, oops. I think the primer might be going that way. Yeah, you might be right. So I don't know if they do anything to stop it, but I think if you try to prime off this end, it's gonna go off it's gonna go off the molecule.
Other thing is.
Oh, no, sorry. No, that's not, yeah. Sorry, this sequence here is the reverse complement of, or is the complement of this, so the primer's not gonna hybridize there.
I know, just looking at this, if you hadn't done that first PCR stuff, you would actually, every time you do nature, just get rid of, so I see that now.
Yeah.
Yeah.
Although it would only matter for the first cycle, so
You would bind and then you'd make a loop and that would just float away and you'd unbind. And you'd make.
oh yeah, that's, yes, you're right, yes. So yes, definitely need to do that. There's also like here, it does, and again, the slide's old, I'm sure that it's changed. There's different polymerase used here. I'm not sure why, maybe it matters, I don't know. I haven't bothered to look up that detail. Other questions? Okay, and the other thing that I'll point out, now actually I'm gonna get to the next slide. So here is looking at a flow cell and you can see like one cycle and you can see it colored based on, the actual fluorescence that you're getting. And one thing that you'll notice is that you could have noticed this on the other one. I feel like it's a little easier to see here. You have spots like this that are well separated from the other spots. And you have spots like this, not well separated. Like here, you can look at it and be like, okay, well, that looks like a yellow and that looks like a red. But you have things like this that you're like, well, are those two spots or one? Here you can get decent separation. Like here is this like one big blobby yellow spot. So because the flow cells, the landing is random, The big limitation on how much data you can get off is you have this balance of, so the more spots you have, the more data you're getting off the flow cell. But if you overload it, then the more you load, the more you get these spots that are close to each other just by random chance. And if they're too close, you're basically going to have to throw out the data because you're going to go back to that problem that I showed you before with the overlapping Sanger chromatograms. You're basically going to get 2 data points from each spot, and you're not going to know what to make of it. So anyway, like you end up throwing away some just by random chance. You're going to have some spots that are too close to each other. But the more densely you load the flow cell, the more that problem. So there's this balance of like, you want to get as much sample on there as you can to maximize the data without tipping it over to you're throwing away too much data because it's uninterpretable. Any questions about that? Okay. So now we've covered most of like why we need to do library preparation. So let's talk a little bit about library preparation. I showed you this like really high level view of library preparation, right? You start off with a purified nucleic acid and you end up with fragments where the adapter is attached to the end of the fragments. So I'm gonna give you like the maybe now 8,000 foot view instead of the 10,000 foot view. I'll talk really quickly about DNA-seq and then I'll tell you how it's different for RNA-seq. And this is all sort of, at least for the DNA-seq and RNA-seq, it's shotgun, which I think I'm gonna explain in a second. Okay, so we start off with our purified DNA. The pieces of DNA are gonna be like pretty big pieces. We need to get down into that 200 to 1,000 base pair range. And so we're gonna fragment the DNA. And there's the most common way to do this is actually physically fragment it. You shear it with sound, and it's like relatively high frequency sound. And there are devices that are tunable such that you can actually like control to some extent. You end up with a distribution of sizes, but you can kind of control what that distribution looks like to some extent, right? So you can really get most of them in that, most of the fragments in the like 200 to 1,000 base pair range. You can do it with DNA. You can also do it enzymatically. That is less popular because it's not purely random. Although companies that sell enzymatic fragmentation kits, you know, claim that they're good enough. I don't know. But the physical shearing is the most common way. So you end up with this like distribution of fragment sizes. Ultimately, it's not perfect. You end up with some stuff that's too big and some stuff that's too small. And so usually there's like a size selection step. I'm not going to go into details about how that's done. And we can talk later if you really want to know. But basically, like these really big things and the really small things get thrown out. And so you end up with like a narrower distribution. And then I'm going to say it's like kind of like magic, but basically you end up, you throw in the adapter, you throw in a DNA ligase, and the adapters get stuck on the ends of the fragments, and you have your library. I've glossed over lots of details, but hopefully this gives you like kind of an idea. There's like a few other steps in there that are important, but not really worth spending too much time thinking about. Questions about that so far? And so I just want to point out, I've kind of said this again, the size of the fragments is relatively random, and the breakpoints in the genome are essentially random. And another thing to keep in mind that I think is easy to get lost is that you're almost always putting in billions to trillions of copies of the genome into this, like at this purified DNA. And so it's not like you're taking one genome and breaking it up randomly. You're taking trillions of genomes and breaking it up randomly so that all those breakpoints are going to be different, right? And so if you sequence it enough, you're going to like have you'll sequence these fragments from all over the genome, and you end up having, depending on how much you sequence and how big the genome is, you can have, sequence each base on average 50 times, 100 times, 200 times. It just depends on how much sequencing you do.
Do you use two different kinds of ligases to get each strand to have different adapters?
Oh man, I was afraid you were going to ask me that. No, to make sure that you have the different adapters on each end. So it's actually a trick that I think I used to have this slide in there, and then I was like, it's too complicated. So it's the way they design the adapters, it's actually for part of it, they're complement for the, let me think, for the, well, I guess it's 3' end of one. So it's complementary like here, but this part is different. And so, so it's basically two oligonucleotides that you can make it yourself, or you can buy it from Illumina or from other places. So you basically, they've hybridized these adapters that I'm going to draw it. And I'm not even going to remember what it looks like. So it's actually called a Y adapter. But this is 5', 3', and this is 5', 3'. So you actually end up with this hybridized. So there's one adapter, but this gets hybridized on here. I'm going to scoot this so people on Zoom can hopefully see it. Yeah. Okay. And if I actually remember how this works, you're gonna, there's a reason why I erased it. And I'm too stupid to remember. Okay, third time's gonna be the charm. What colors can I use? Purple. I'm tempted to do green and orange, but that is not 5', 3', 5', 3', and then so this is 5', 3', 5', 3'. Okay, so then on this end, it looks like this, and see, now you're regretting that you asked the question. I don't know if I've ever actually been asked this question. Like I said, I think I used to talk about it. I was like, it's probably not worth it. Okay, this is 5', 3'. Okay, so that's what it looks like when it's initially hybridized. And then once you denature it, now one of the strands is going to be like that. Right, so like you have this little bit that's complementary here, but at this end, which is 5' and 3', depending on which way you go, so you end up with single-stranded fragments that basically have a different adapter. I'm glad I actually remembered how that worked. I think I'm not lying about this. I'm pretty sure that's how it works. But the best part is that it's called a Y adapter because it's. And actually, there are other ways to do it. There's another. So this is. I'm pretty sure Illumina has a patent on this. There's another company that actually has these hairpin adapters that like this is the 3' end and this is the 5' end, and it's like a single thing. And that they like claim that, you know, it helps. You get this like adapter dimerization, so they claim that it helps with that. Anyway. It's 3, maybe we'll take like a 5-minute break, how's that? Well, let's take a break till, it's 3:03, let's take a break till 3:10. Nice round number. Okay, I'm gonna get going because I've got like 40 slides to cover in the next 50 minutes. That shouldn't be a problem. I should have just said it's magic. That would have been faster. That's okay, right? Okay, questions about this? Okay, so let me tell you like subtle differences between RNA-seq. And just to be clear, this is like sort of shotgun standard, like for the DNA-seq, like grind up something, extract the DNA, right? We're not doing it one of those like other 59 things that you can do with DNA-seq that's on the Illumina poster. And the same thing with RNA-seq here. So like major differences, so like short summary of what you're doing with DNA-seq. So major differences with RNA-seq is that number one, you're gonna be purifying RNA, not DNA. And number two is for at least Illumina sequencers and most sequencers, you can't sequence RNA directly. You actually you can only sequence DNA. And so you have to make DNA from the RNA. That's the major differences. There are some details we can get into. You can also, like the fragmentation ends up being a little bit easier with the RNA because you just like throw a divalent cation like magnesium into the RNA and you heat it up for a little while and it will, because RNA is auto-catalytic or can catalyze its own degradation as long as it has a divalent cation, it just will fragment itself, which is nice. Questions about that? Okay, yeah, so I'm glossing over some details, but that gives you the basic idea. And then, so for amplicon library prep, in a way, it's even easier because you kind of like, usually you can skip almost all that stuff. You just purify the DNA, and then you do a PCR where the PCR primers have the adapter stuck on the end. And I'll show you exactly what I mean here, although that could be a little bigger. But basically, like, here's the PCR primer, and here's the other PCR primer, and that's the adapter, right? You've actually synthesized the PCR primer with the adapter at the 5' end. And so when you do the PCR, you end up with the adapter is like sort of ligated on in the same step. I mean, it's not ligated on, right? You chemically synthesize the primer with the adapter as part of it, or the 5' end.
These randomly generated primer sequences.
So if you're doing amplicon sequencing, then no.
Oh.
You're like targeting one or several regions of the genome, right? And so in this case, so like for example, for most common application I see, which is 16S ribosomal RNA sequencing, right? You design your primers to a specific region of bacterial 16S ribosomal RNA, and you're just sequencing that part out of all the bacteria that are in your sample. So like one relatively important detail about amplicon sequencing versus shotgun sequencing is that for an amplicon library, the fragments are all almost exactly the same size. It depends on exactly what that amplicon is because you can get size variation, but like for 16S ribosomal RNA-seq, the amplicon that people normally do, you get size variation between like say 245 base pairs and 253, which is like pretty minor variation. For shotgun sequencing, the size of the fragments are totally random. And so you get this, you know, you're trying to get in this range of 200 to 1,000 base pairs. But you get things smaller and you get things bigger. And even with the slide selection is still stochastic, so it narrows that window, but you still get stuff that's smaller than you want and stuff that's bigger than you want. Questions about that? Okay, so I've kind of already given you most of the answers for this, but why do we need adapters? There's a little bit of review, and I think I have one or two new things I haven't mentioned before. I have a hint for you, which is, oh, okay, I got an answer in chat on Zoom, which is for multiplexing, which is the one thing I haven't mentioned. But yes, although you only need it for multiplexing if you want to multiplex. I'll talk about multiplexing in a second. Okay, so here's the hint. So why do we need adapters? Specifically, what's the, huh? When? Yeah. What's that step called? Yeah, the bridge amplification. Well, there's that initial copying, but yeah, the bridge amplification. So in that situation, what is the adapter doing for you? Well, yes, kind of, but it's a priming site. Okay, so what else do we need the adapters for?
Find the flow cell and then the primer site.
Hybridizing to the flow cell. Primer site for bridge amplification. There's one other thing that I've mentioned. Maybe I'm like cheating a little bit here because you kind of already said it, but not exactly. So you actually need priming site for two different things. You need it for the bridge amplification and for the sequencing primers. And actually it's actually different parts of the primer. It's the 5' end is for the bridge amplification, and then like more 3' of that is the sequencing priming site. Because for the sequencing primer, just here, you just want to prime starting right there, because if you prime starting back here, then you end up sequencing some of the adapter itself, which you don't care about. So you want to be priming right at the junction. Whereas for the bridge amplification, you need to kind of amplify the whole adapter. Actually, I don't know, I guess the primers on the flow cell, the oligos on the flow cell could be the full length. No, they're not, and I know why. I can tell you in a second. Okay, and so that's the answer. And then the other thing is what was mentioned in the, someone said annealing. Yes, so hybridization or annealing. So, and then the other thing which was mentioned in the chat is index barcodes for multiplexing. Okay, so what's multiplexing? Again, probably people have heard of this, but I'll just review really quick. The idea of multiplexing is that you can put multiple samples in a lane, and it lets you, so the reason for doing this is because these things generate a lot of data, and it's relatively expensive to sequence, although the price has gotten crazily cheap. But if you could only put one sample per lane, basically the sort of capacity of a flow cell has exploded. The cost of sequencing for a given flow cell, I'm not going to say it's been constant, but it's sort of like the cost per base pair has come down, but the cost per flow cell I don't think has really changed that much. And so you wouldn't necessarily, for most applications, you don't necessarily want a whole flow cell or even a whole lane for a sample. You can put, for a lot of applications, you can put 100 samples on a lane or more. Like sometimes you can put 1,000 samples on a lane. But in order to be able to do that, you need to be able to, so that's multiplexing, is putting multiple samples on a lane. You need to be able to figure out which sample a given sequence came from. And so for that, you need barcodes. And so barcodes are a little bit of sequence that is not biological. It's an index. It's a way to distinguish different samples. And so what you do is when you're preparing your libraries, for a given sample, you assign it to a barcode. And then at the back end, when you get your data, for every biological sequence you get out, you also get the barcode that's associated with it. So then you can do what's called demultiplexing, which is assigning a given read to a given sample. And so even though you threw everything on one lane, you can then like deconvolute that into individual samples at the end. And so here's one, this is actually an old slide, but I like it because it's a little bit simpler. So this is the structure of, again, it's a little bit out of date, but this is the structure of what a flow cell looks, or what a fragment looks like. We've already seen this before, but here it zoomed in a little bit. So here's the biological sequence. Here's the adapter at one end. And like I mentioned, the green part here is actually what's being hybridized, and the complementary sequence for the oligonucleotide flow cell that serves as the priming site for the bridge amplification. Here's the sequencing priming, the priming site for the sequencing primer. And then on the other end, actually I haven't talked about this yet, but you can prime off this end. Here's where the barcode index sequence goes. And so it's stuck here usually between the sequencing priming site and the amplification priming site. So these days, it's pretty common to have actually have dual indices. So you would have an index sequence here and then a different index sequence here. And so I did this, I should have done this in a different order. Oh, well. We'll talk a little bit in how you actually get the, oh, you don't see the mouse in the slide. Oh, that's because I have it in the wrong place. Sorry. I was pointing at something on my computer. So here's the sequencing, the index primer here. And again, that if you have dual index primers, you would have a separate index sequence here. I'll talk in a little bit how you actually get this information, because I maybe should have done this slightly different order. It's good. But the way that you do this, like from a practical point of view, is that while this sequence here, this sequence here is the same for every single fragment, and this bit are the same for every single fragment, this is going to change for different samples. And so you actually get, for every sample, for every index that you have a different, if you just have single index, if you just have a barcode index at this end, then you're going to use the same adapter for all your samples on this end, and just a different adapter for each different sample on this end. If it's dual index, then it's actually different on both ends. And so what you do is you just make sure when you're doing your library prep, you know, back here we said, okay, treatment B, replicate 3, we're going to give it this sequence as a index barcode. So then you just make sure that, I have different sample names here, but for sample A, you put in the adapter that has barcode 1 in it. For sample B, you put in the adapter that has barcode 2, right? And just, so when you're doing your library prep, that's when you're sticking the adapter on. Then later on, you're going to pool all the different samples once you've actually got the adapter on, and throw all that on the flow cell for sequencing. Questions about that? Okay, so I have even more details. Okay, so first let's talk about how reads are structured. or how, like the kind of, what's it called? There's probably a good name for it. Sequencing strategy, I guess. So far what we've talked about is what you would call single-end sequencing. And that means that when we kind of touched on this before a little bit, when you're sequencing, the sequencing primer that you're using, you're priming off this adapter, so you're sequencing from this end. Now, I kind of glossed over this before, but there's also a priming site, sequencing priming site on the orange adapter. And so if instead of using this primer, you throw in this primer at the beginning of the sequencing, then you can prime off this adapter and you'll sequence that end of the fragment. So that's called paired end sequencing. And that's like, it's a little bit confusing. So if you're doing 50 base pair paired end sequencing, you're going to throw in the primer that's for this site, on this side. You're going to hybridize it. You're going to do your 50 cycles. And then there's like a little bit of chemistry that you do to denature it and get rid of that. I think there's something else that goes on, but I've never bothered to like figure out exactly because I don't care that much. But basically after you've done that, you kind of clean stuff up. I think that actually what you end up doing is you end up like synthesizing the other strands. I'm not sure about that. Anyway, I looked at it at one point, I was like, I don't care. So you put in the primer that's complementary to the orange adapter, the sequencing primer that's complementary for the orange adapter, and then you do another 50 cycles going the other way. That makes sense? So you get the specificity basically by that sequencing primer. So yeah, so you're gonna get your like whatever cycles you're doing, whether it's 50 cycles, 100 cycles, 150 cycles, you're gonna do off that end. And then here I'm not showing it how it happens, right? And then you're gonna denature that and then throw in the other adapter in a sequence this way. Does that make sense? And so like here's what you would get one way, you get this complementary sequence. From the other end, you're gonna get this sequence. Now, what you'll notice, what I did here is like, 9 base pairs? I think I have to look the cost up again. What you'll notice is that there's a whole region in the middle here that I'm not getting a sequence from. And that's actually pretty common. So, for example, wait, I'm confused. Did I just have that slide twice? Okay. So, for example, let's say that I'm just looking at one fragment, the insert size, I don't know if I've used this term before. Insert is the biological DNA, the black stuff. So the insert size here is 273 base pairs. I'm doing 50 base pair single end sequencing. So I'm going to sequence 50 base pairs from the left side here, and I'm going to have 223 base pairs of that fragment that I'm just not going to sequence. I don't know the sequence of it. That's just the way it goes. If I was doing 50 base pair paired end sequencing, I'd have 50 base pairs from this end, I'd have 50 base pairs from this end, and then I have 173 base pairs in the middle here that I don't have a sequence from. That's how it happens. So if I have a long enough read length and my fragment is small enough, I can overlap the reads, right? So here's 250 base pairs. Why did I say single end? Because I have it. Oh, well. If I was doing single end, this would be 250 base pairs, right? There's still 23 base pairs at the end that I have sequence for. If I did paired end, I messed up this slide. If I did paired end, I'd have, they'd actually overlap for most of this. Now, because remember that for every base, we know the previous base, for every cycle, we know the previous base because it came from the same spot, right? Remember that? So that's how we know that we're going to get this sequence and we're going to get this sequence, and we know that they came from the same fragment because they came from the same spot, even though it's like a separate set of 250 cycles. right? So even in this example, where I have a 618 base pair insert, I'm doing 250 base pair paired end reads. There's this bit in the middle that I don't have a sequence for. I still know that this is the sequence from the left end, this is the sequence from the right end, and I know that those came from the same fragment. And I know roughly how far apart they are, because I know that the fragments have to be between about 200 base pairs and about 1,000 base pairs, right? So they shouldn't be much more than about 1,000 base pairs. You know, the stuff in here, if I have 250 base pair paired end reads, this gap in the middle shouldn't be more than about 500 base pairs, right? It's not, that 1,000 base pairs isn't like a firm number, right? So it could be, let's say, you know, maybe it's 1,000 base pairs in the middle, but I have some rough fix of the relationship between these two in terms of like the genome distance. Make sense?
Complicated math.
Say that again?
Some complicated math that would put that knowledge together. Overlapping things.
So it depends on what you're trying to do. Sometimes you don't care, and often you don't care what the distance is.
Treat each like its own shot.
Yeah, or if you, it depends on what you're doing, right? So if you're like doing RNA-seq, then you're just taking it all and you're finding where it came from in the genome. And so you don't have to you don't really have to figure out like you can actually then back calculate what the distance was, what that gap size was, because you know, like, well, this went here and this went here, and in the genome, they're separated by 600 base pairs, right? And so the original insert size must have been whatever, you know, 600 plus whatever the read length is.
What would be the point? What would be the benefit of doing head and plus single end? Is that
so it depends. So it depends on what your goal, what the goal of the sequencing is. So, and there's kind of a question in there that you're not asking, but you are asking, which is, so basically the longest reads you can get on Illumina are about 300 base pairs, but you can do paired end sequencing and do 300 on one end and 300 on the other. And so that limitation of 300 base pairs is that the quality goes down as you go from the 5' end to the 3' end. And so if you looked at a graph of the quality, it's gonna go down, like I said, from 5' to 3'. When you do paired end sequencing, you kind of reset that. It's not exactly, it doesn't go quite up to the highest quality, but it's much improved from, like, the quality of the first base here is gonna be a lot higher than the quality of the last base here. So it basically allows you to get from a single fragment. You couldn't do a 600 base pair read on Illumina, but you can do 300 plus 300. So that's one reason, is that it just lets you get more sequencing. And usually the cost of, say, at least the pricing that they do at the sequencing core here, the cost of usually of 100 base pair single end is going to be a little bit higher than the cost of 50 base pair plus 50 base pair paired end. So that's one reason. Another reason that it's beneficial is if you're doing de novo assembly, the fact that these fragments you have sequence from like both ends of the fragment, even if you don't know what's in the middle, you're kind of getting a little bit more coverage of the genome. And one of the tricky things about de novo assembly is like kind of putting the pieces together. And so the bigger the gap that you have, you actually, if you're doing de novo sequencing, you can never get the whole genome, even for like small, I mean, for viruses, I guess you can, but like even for like bacterial with small genomes, no one's ever been able to get a full, complete genome assembly with just paired end sequencing. You actually have to do this, there are a couple of different ways to do it, but you basically need to make a different type of library that gives you bigger gaps. And so it helps make those connections over longer regions, just because they're end up being parts of the genome that you can't like sequence, where basically you might have thousands of base pairs of repetitive sequence that you just can't get across with paired end sequencing. But it does, having paired end sequencing does help you kind of like get a little bit of a wider coverage than you could just from having single end reads. There's some other reasons, like for example, here where you have this overlap, I told you that the error rate for Illumina, at best, you're getting about 1 in 10,000 chance of error, which sounds great, like I said, until you realize you have billions of bases of data. So when you have a lot of overlap here, when you sequence, you basically sequence most of the bases in this fragment twice. And so that actually, you can think of that as 2 separate measurements, and it improves the accuracy. I'd have to check on the math, but it's roughly, 1 in 10,000 times 1 in 10,000, which is, what is that? 10 to the 4th times 10 to the 4th. So it's like 10 to the minus 8, right? Which is 1 in 100 million error rate, which is a lot better than 1 in 10,000. I'm not sure that the math works out quite that way, but it's, you do get this multiplication of this improvement of the quality. So those are a few different reasons. There, I'd have to think if there are others, but there are several reasons. Other questions?
Can you basically, during your size exclusion, can you limit the length of your transcript? Instead of like having them go over, say, 300, can you just put it in another one?
So you're never gonna, you always get a distribution of sizes. It's a good question. Basically you bring the distribution in, so it's more peaky, but it's still a distribution. You do have some control over like how broad that distribution is, but it's not like perfect control. Is that answering your?
How do you do it?
So usually so there are kind of 2 ways. The sort of old style way was you'd actually run the library on a gel and you'd actually cut out bands and then extract the library just from the band. The way that you can imagine that's time consuming, and like you'd never do that if you had like hundreds of libraries. So the way that it's done now is with what are called spry beads. I'm trying to remember what they're actually made of. I don't remember. I think it's like basically like kind of it's like maybe it's silica. And so the thing about spry beads is that you can control, you add your sample to the beads, and the beads are like in a solution. You can see they're like not quite microscopic. You can see them, but they're like close to being microscopic size. And so the beads are suspended in a solution of polyethylene glycol, and you add your library to the beads or your DNA. Usually you actually make the library, if I'm remembering right, you make the library first and then you do the size selection. I think that's right. It's been a long time since I've done it. So you add your sample to the beads, and you can control what size range of fragments stick to the beads based on the concentration of, I think it's the concentration of the polyethylene glycol. And so basically by adjusting the concentration, you can kind of like get the sort of everything below 1,000 to stick to the beads. And so then you take everything that's in solution, you basically take the solution and you throw that out. So now everything that's 1,000 and smaller is stuck to the beads, and everything bigger than 1,000, you throw it out. And then you switch that up and you change the concentration. So now stuff that's bigger than 100 goes into solution, stuff that's smaller than 100 or 200, whatever you choose, is stuck to the beads. And so you take now what's in solution, and that's bigger than 200, but smaller than 1,000.
And the risk in trying to go narrow up is you're losing like.
I think that you can try and go narrow. You can try and go narrower. It's just that you're never going to get it. Because it's not perfect, right? It's not that you're guaranteed that everything stuck to the beads is below 200. Yeah. So usually you're aiming for like, say, like a peak of about 300. Sometimes you'll aim for like a little bit higher, but you really do get a pretty broad distribution of fragments, even with that. It's just that you bring the like peak size, you make the peak sharper. Other questions? Okay, so let me tell you really quick, this doesn't cover everything, but most of what I've told you today is basically the technology is the same again. You'll hear about the different platforms that Illumina has, MySeq, NextSeq, NovaSeq. This is all pretty much the same across them. And the big difference is, there are a few technology differences that I can tell you about in a minute, but the big difference is if you look at the surface area of the lanes on the flow cell, the surface area is basically proportional to the amount of data that you're going to get out. So this is a NextSeq, this is a MySeq. You guys can probably all see the lanes from there on the NextSeq. I'm guessing you can't see the lane on this one because it's so small. So you get a lot less data off the MySeq. So running the MySeq is cheaper per run a little bit, but per base, the NextSeq is a lot cheaper. And the NovaSeq is even cheaper than that. So really the big differences are like the cost, the amount of data you get, and the cost per base. Like I said, there's subtle technological differences. And you can see that even more so here. So these are all NovaSeq 6000. There's actually now a newer NovaSeq out, but I don't have a picture of that. But you can see here, these are the different NovaSeq flow cells. And if you look really carefully, first of all, this one has 4 lanes, and these guys all have 2 lanes. But if you look more carefully, you'll see that the lanes on this one are a little bit wider than the lanes on this one, and the lanes on this one are a little bit wider than the lanes on this one. And that's just changing the surface area. So it just changes the amount of data you get off. It does change the runtime also, because actually a big part of the runtime is imaging, because that confocal microscope, it can't image the whole lane at the same time. It actually like takes an image and it scoots over a little bit and takes an image. So it actually images the lane in tiles. And so that's actually a big time consuming part of the sequencing. Okay. I have a link in here for a video that like goes through all this stuff that I just told you over the past almost 3 hours in like 3 minutes. Hopefully I did a better job of explaining than the video, but maybe not. Okay, so I told you there are some subtle differences between the different flow cells, and I also told you that I lied to you a little bit before. So the newer machines, so NovaSeq 6000, I'm 99.9% sure for the NovaSeq X is the same. Instead of, what I initially told you is that the way the library ends up on the flow cell is like throwing jelly beans on the ground. And that's how, let's see. Yeah, this is on here. So this one and this one, that's how they work. For the newer machines, they have what are called pattern flow cells, and they look like, if you do a scanning electron micrograph, this is what it looks like. It's like these little wells that are patterned across it. And so for these, the fragments can only land in the wells. The other part of the surface here, I think that it doesn't have the oligos, although I'm not 100% sure that's how it works. And so instead of randomly scattering, you get this regular grid of spots. And so that actually improves the density at which you can load it. Because remember, the limiting factor of loading these is that if I throw too many jelly beans, you're going to have jelly beans that are right next to each other and I can't distinguish them. So the whole reason for doing this is that it improves the physical separation, which enables you to resolve the more spots. So you can essentially saturate those wells. It turns out that the chemistry works a little bit differently. I can talk about that if anyone cares, but it's not that important. So another difference, and this is, I think this is for the MySeq. It used to be for the MySeq and the older machines that each base had a different color fluorophore. So it meant that you actually had to capture 4 different images of each spot, right? So I told you that it actually has to scan the flow, scan the lane, and capture a bunch of images. It actually would capture 4 images in 4 different channels. So for the newer machines, including the NextSeq, instead of having 4 different fluorophores, they only have 2. The advantage of that is that with 2 different colors, you only have to capture 2 images of each spot. So it's half the imaging time, which, because the imaging is like a big part of the time it takes to run a sequencing run, it actually sped things up a lot. And so the weird part is that there are 4 bases, and you think like, okay, well, with 2 different colors, how do you distinguish the 4 bases? So T is green, C is red, A has a red and a green, and G doesn't have anything. Which like people were super concerned about because they're like, well, you're saying no data means something. It ends up that the people were concerned that the data quality wouldn't be as good off these, but I think that it has ended up being roughly as good. So that's the big sort of details that I glossed over with that. The newer machines have those differences. Questions about that? Before I get to talk about like one of my favorite pet things, I include these other slides. Okay, so I want to talk really quick about the 3rd generation technologies. And there are like lots of different cool aspects of them. One is that the read lengths are like huge potentially compared to the Illumina. So wait, do I have the table? There it is. So this is a really old table that I haven't updated. So Nanopore, I think people have gotten up to like 2 megabase reads, and PacBio, I think the number is bigger here too. But you're talking about read lengths of tens of thousands to 1 million base pairs for these technologies, whereas remember on Illumina you get 300 base pairs. So there are pluses and minuses of these other technologies. Long reads are really nice. The cost per base is a lot more expensive on PacBio, and the number, like I said, these numbers are really old. But the cost per base is still a lot more expensive on the 3rd generation technologies than Illumina. The raw quality per base is a good bit lower. So these numbers are also old, but it's still like the best you can get sort of raw quality per base on Nanopore is about 99% accuracy, or like 1 in 100 error rate. There are ways to compensate for that. But like I said, there are pluses, like you get these really long reads. I can go back and talk a little bit about the benefits of that in a little bit, but I want to talk about the technologies. So where's my bag? So let me talk about Nanopore first. first because special love for it. I'll pass this around as long as you promise to be gentle with it. So this is a nanopore sequencer. This is the flow cell. This is the sequencer. If you look at it, it's got like a funny, I don't know what it's called, but it's basically the USB. It's not C and it's not A or B. I don't know what it is, but like I have a hard drive that has this port. The other end of the cable is just like a regular USB and you plug it into your computer. You'll see that it's relatively small. And this picture's old, but there's that nanopore sequencer. This is an old HiSeq 2000, so it's like the newer machines are like about refrigerator size. Actually, I'm exaggerating a little bit. A MiSeq is like kind of microwave size, like about that big. But the NovaSeqs are like about like kind of small refrigerator size. This is the old PacBio machine. The newer ones are about the same size, about the same size as the NovaSeq. Let me tell you really quickly how the nanopore works because I think it's super cool. So nanopore is the only, as far as I know, it's the only technology in the market today that doesn't use any imaging. The way it works is it has these nanopores in a membrane, and the DNA, there's like a protein that attaches to the DNA, and it gets fed through the pore, and there's a charge across there's an electrical current, there's a charge differential across the pore. And so because of that, you get current flowing, you get ions flowing through the channel. And you can measure that with a very sensitive ammeter. You're basically measuring the flow of electrons or ions across that channel. And so the size of the pore is just a little bit bigger than the size of a single strand of DNA. And so the DNA, single strand of DNA will fit through that pore. But as the DNA is going through, the bases are in the backbone are to some extent blocking the pore. And so they change the rate at which ions and electrons can flow through the pore. And that changes the instantaneous current that's flowing through that pore. And so what you're measuring with nanopore is actually the current as a function of time. And then those current measurements get fed through a deep learning model that's been trained on known sequences. And so you can then call the bases based on that current change. And actually, for nanopore, you actually get base calling in real, or you can get base calling in real time. So you can imagine this is part of the reason why the data is noisy and like not super high accurate is that you're measuring this current and feeding it through this deep learning model. For PacBio, which is another cool technology, but you can't pass it around because, like I said, the machines are like that big. I will say as an aside, nanopore is the only sequencing technology that's been used in space. They took one of those on the International Space Station and did sequencing, which I think is super cool. So PacBio, it does use it does use sequencing by synthesis. It is also, oh, actually, so sorry. So for nanopore and PacBio, you're actually getting single molecule data, right? So you're actually getting the sequence, whereas for, Illumina, really each spot is millions of molecules. For nanopore, you're getting data from each pore, separate data from each pore, and at any given time, a pore has one DNA molecule running through it. Similar for PacBio. PacBio, you have these micro wells, kind of like the micro wells on the newer Illumina flow cells. And there's a polymerase stuck at the bottom, a single polymerase stuck at the bottom of the nanowell. And actually, this is not the best representation because the DNA is usually, it's a single-stranded loop. And so here, the DNTPs, they're not terminated, but they do have, remember that pyrophosphate, that's the two phosphates that go away once the base is added. So on the pyrophosphate, they have, that's where the fluorescent dye is. And they have some like super fancy optics going on that I can't even begin to understand. It's called like a zero mode, oh there, it has a zero mode waveguide, such that it has two cool characteristics. One is that it's super sensitive. And so it can actually, unlike Illumina, it can actually detect single fluorescent molecules. And it only detects fluorescence within a very narrow distance from the bottom of the, they don't call it flow cells, they call them smart cells, but same idea. So it can only detect, basically do imaging within a very short range of the surface. And so what happens is you get, here's the data, and it's fluorescence as a function of time, and you can see it's like kind of noisy. And so what happens is that you get the nucleotides with the fluorophores kind of just by Brownian motion popping in and out of the range of the imaging. But as a base is being incorporated, you can imagine the Brownian motion is like, the fluorophores are like going all over the place on the scale at which the imaging works. But as the base is being incorporated, it's like kind of like stuck there by the polymerase for, you know, a millisecond, but the rate at which it's moving compared to everything that's like free, it's like stuck in molasses. And so during that time that the base is actually being incorporated, you get this signal peak of that in that channel of that base. And so so you're basically imaging the nucleotide as it's being incorporated. And then because the fluorophore is attached to the pyrophosphate, once you get the cleaving between the first phosphate and the second and third phosphate, then the dye diffuses because it's no longer attached. It's no longer in that DNA chain, and then another base can get incorporated. So it's a super cool technology. Again, but one of the reasons why the accuracy is low is because you're looking at single molecules, and the polymerase just has an error rate to it. But there are ways that you can analyze the data. You basically stack up multiple passes on this molecule. Because this is a loop, the polymerase is gonna go over it several times, and so you actually get multiple samples of that data. So anyway, it's a cool technology. I think I will shut up now because I've got like 1 minute left, and I'm supposed to ask you all to fill out this survey. It's in the, if you're on Zoom, the link is there. If you're here, then the link is there. So please fill this out. Ideally, if you could just take like, I think it should take like 3 minutes. If you could do it now, that's great because it helps us, you know, plan for future sessions and also for next year. And if you have questions, I can answer questions. Oh, actually, there was one question I think I saw in the chat. So there's a question in the chat. For small sequences like microRNA or other non-coding RNA, do you suggest single or paired end 50 base pair sequences and why. So it's a good question. So if you can, what you would get out of paired end sequencing is that you would, because usually for like microRNAs, it's 23 base pairs, and the 50 base pair read's going to cover it and then actually read into the adapter. You are going to, the paired end reads doesn't buy you anything in terms of like getting more sequence. What it does give you is it'll give you more accuracy, right? Because you're sequencing that fragment twice. And so if that is valuable to you, then yes, there's value in doing paired end sequencing, but it's hard to know without knowing any sort of more detail whether it's valuable, right? So if I mean, you have 23 base pairs, if there's, you know, if you're working with, say, the human genome and most of the microRNAs are, you know, have a few, I don't actually know this off the top of my head, but if you have, you know, microRNAs if they don't tend to like be within like 1 or 2 base pairs of each other, which I would imagine most aren't, in terms of sequence, like what, you know, one base sequencing error isn't going to change which microRNA you're thinking of, then maybe it doesn't matter. But that's something I don't know off the top of my head. It's been a long time since I worked with microRNA data. Last time I did, I think it was single end, but it's been a long time. Other questions here or on Zoom and it is 4:02, so if people need to leave, feel free to leave.
Thank you. Thank you.